So we've already talked about how gene regulation is accomplished in prokaryotic cells at the level of transcription by repressing or activating expression of, for example, the lac operon using transcription factors like CAP or the repressor protein to either activate or repress expression of the operon under different environmental conditions. So that's a really typical mode of gene regulation that we see happening in prokaryotic cells. What we want to do now is turn our attention to eukaryotic cells, which, as we've talked about many times before, are much more complex in terms of their genetic composition. They're more complex in terms of their cellular structure, and likewise what we're going to see in terms of gene regulation and the regulation of expression of genes in eukaryotes is that that's a lot more complex as well. And there are many mechanisms for regulating gene expression that are available in eukaryotes that are not in prokaryotes. And we've talked a little bit about this before. For example, when we talked about transcription and translation and discussed alternative splicing, that's definitely one way that we can regulate the product or expression of a gene at the level of the mRNA, and that happens in eukaryotes and not prokaryotes. All right, so what we're talking about here, just to refresh our memory and get on the same page here, is we're talking about genes in the genome, genetic instructions to the cell for how to synthesize either a protein product that's going to be encoded by an mRNA intermediate, as we've talked about before, or a functional RNA molecule. We know that all of those types of gene products are encoded by genes, specific sequences in the genome. So when we talk about regulation of gene expression, what we're really talking about is regulation of the production of that product. Now, usually genes do encode proteins, so we're going to focus on that. But when we say regulated gene expression, what we're really saying is the protein there or not? Is it functional or not? And in eukaryotes, that regulation can happen at multiple steps along this progression that we see occurring between transcription of the gene into mRNA and translation of the protein at the ribosome, and even beyond that, regulation of how long the protein sticks around and what its structure is. So we can sort of describe multiple steps at which eukaryotic genes are regulated, all the way from transcription of the gene down to degradation of the protein to get rid of it from the cell. Now, if we think about regulated gene expression from a larger perspective, from the perspective of the cell, and ask ourselves, for example, in a human cell we have 20,000 genes. Would we expect all of those genes to be regulated in their expression? Or would we expect that there would be some genes that are not regulated, in other words, constitutively expressed? So can you think of any proteins or gene products that we've discussed in the course so far that might be important enough for the life of any cell that it would have to be expressed all the time and essentially not regulated? And if you think about it for a little while, you'll see that some of the basic cellular processes that we've discussed, for example, in our metabolism unit, you can see that a cell is pretty much always going to have to be expressing those proteins, right? So, for example, the glycolytic enzymes, proteins that are in the electron transport chain, things like that. So those are referred to as housekeeping genes or housekeeping proteins, because they're carrying out basic biological functions. And so those would be what we would call constitutively expressed. In other words, not regulated in their expression. Their basic biological functions that they're carrying out have to be carried out all the time in all cells. So things like, components of the cytoskeleton. Do we always need to make tubulin protein so that we can create microtubules? Yes, absolutely. So those types of things. How many genes out of the 20,000 would fall into that category of housekeeping genes? About 4,000. Estimates vary. But aside from those, the other, 3/4 or so are regulated in their expression. In other words, in some cell types they may be expressed, in other cell types they wouldn't be expressed. Perhaps they're expressed in a given cell type under some environmental conditions or situations and not in others, but regulated in some way. Right, so the vast majority of the genes in our genome are regulated in their expression. Now, of those genes that are regulated, about 70% of that regulation comes in here at step 1, transcribing the gene, right? Do we transcribe the gene to make the mRNA or not? That's approximately 70% of the regulation that goes on in regulated gene expression. Now, in addition to that, though, we have all these other mechanisms by which we can regulate the expression of a gene and whether a gene product is present in the cell or not. We know that alternative splicing can lead to different proteins from the same gene by splicing together those exons in different patterns, removing introns in different patterns. But we haven't talked much about the fact that exporting this mRNA from the nucleus is an energy requiring process. It's actually regulated, and so some mRNAs will be transcribed from their gene, but then they'll be held in the nucleus until a signal is received, at which time they'll be exported. So we can regulate nuclear transport. We have talked about the fact that mRNAs can be regulated in their stability within the cell. Some mRNAs can be rapidly degraded or targeted for degradation, whereas others will have a very long half-life and will essentially stick around for hours and hours and hours. All right, so we can regulate at that stage. As we're going to see in our next discussion, looking at development in Drosophila, many mRNAs are localized. In other words, once they're transcribed, they're actually physically transported to a specific location within the cytoplasm and tethered there. And then finally, at the level of the mRNA, we can regulate the interaction between that mRNA and the ribosome, such that we're regulating the initiation of translation, and therefore we're regulating how often this protein gets synthesized by the translation of this mRNA at the ribosome. Now, all of these mechanisms of regulation, as we talked about very briefly, are going to be mediated by those regulatory sequences that we have in the mRNA, which are going to bind RNA binding proteins that are going to be responsible for regulating these types of events, right? How long the mRNA sticks around, whether an exonuclease gets recruited to degrade it or not, etc. So that regulatory function of the untranslated region comes into play when we're thinking about these modes of regulation. And then finally, once we've synthesized the protein, we have additional mechanisms by which we can control the structure of that protein and how long it sticks around in the cell. Very often, proteins will be processed in which specific amino acids are cleaved off. This is called processing. And so we end up with a shorter polypeptide sequence that gives us our functional product. So we could synthesize a protein, but it might be inactive in its initial precursor form, and only when it's enzymatically cleaved does it become an active protein that can carry out its function. So that's another mechanism by which protein or gene products are expressed. And then we did talk about protein degradation as a means of regulating gene expression. And remember, in our mitosis unit, we talked specifically about the fact that cyclin protein, for example, gets degraded at specific time points in the cell cycle. And you'll recall that that was mediated by the enzymatic activity of the anaphase promoting complex, which added ubiquitin to that cyclin protein, and that targeted it to the proteasome for degradation. So we're not going to spend much time talking proteasome. Yeah. We're not going to spend much time talking about these other mechanisms. Mostly what we're going to talk about in this video is regulation at the level of transcription initiation. And then in the next video we'll talk about mRNA localization and how that can lead to transcriptional regulation such that we get specification of different cell types and an overall body plan during development. But in this video, let's talk about transcription initiation and how it's regulated in eukaryotic cells, because we see some similarities with what we saw in prokaryotes, but we also see some distinctions that are unique to eukaryotic organisms as well. All right, so if we think about a simple question like the fact that we see different functions associated with different cells in the body, right? When you think about that, how does that arise? Is it because these different cell types in the body are actually genetically unique. In other words, they have different genes and different genetic instructions. If we look at a pancreatic cell, for example, specific pancreatic cells within the pancreas are responsible for synthesizing the protein hormone insulin. So there's an insulin gene that gets expressed in the pancreatic cell. If we look at red blood cells in their immature state, we wouldn't see that insulin expression. Is that because the insulin gene is lacking in those cells, or is it some other mechanism? Well, it turns out that it's not that the insulin gene is lacking in those cells. It's that the insulin gene is present but not expressed, right? Likewise, in this red blood cell in its immature state, we would expect to see expression of the hemoglobin gene, such that we'd see hemoglobin proteins, alpha and beta subunits expressed and assembling into that hemoglobin complex that we talked about before. The reason that hemoglobin protein is not expressed in the pancreatic cell is not because the hemoglobin gene is not there, but because it's being specifically regulated in a way such that it's not being expressed in the pancreatic cell, but it is being expressed in the immature red blood cell. Just a note, why immature? Well, it turns out during the maturation process, red blood cells actually get rid of their nucleus. They extrude their nucleus out of the cytoplasm. So red blood cells in their mature state are one of the only cell types in the body that actually don't have a nucleus. They don't have a genetic program still being in play. What they are essentially is big bags of hemoglobin enclosed in a membrane. Okay, so what's responsible for this differential gene expression? Well, as we talked about briefly before, it's going to be the activity of these regulatory transcription factors. Regulatory transcription factors influence the expression of specific genes in the genome, either activating their expression or repressing their expression. Now remember, we talked about general transcription factors, which were required for any gene to be expressed because they assembled at the promoter and they recruited our RNA polymerase enzyme to the gene so that that gene could be expressed. Now those are used for all genes, any gene, whether it's regulated or not, right? Here what we're talking about are specific regulatory transcription factors that interact with regulatory sequences in specific genes in the genome and either activate or repress their expression. Typically what we're talking about is activation in eukaryotic cells, because it turns out that in general the default state is off until switched on. And so most of these transcription factors are going to be activating in their function rather than repressing in their function. And so we could imagine that in order for this insulin gene to be expressed, there are going to be specific sequences in the gene, which are sort of illustrated by these different colored boxes. These would just be specific sequences of nucleotides to which different transcription factors would come along and bind in a sequence-specific fashion. And when those transcription factors bind to those sequences, we see an interaction between those transcription factors and the complex of proteins assembled at the promoter that leads to activation of transcription and lots of mRNA being transcribed from this insulin gene. And so the reason the insulin gene is expressed in the pancreatic cell is because the correct transcription factor population is being expressed that can bind to those sequences and activate the gene in its expression. Why is the hemoglobin gene not being expressed? Well, we may see expression of one or more transcription factors, but not all of them that are required to activate expression of the hemoglobin gene, right? So 2 additional, let's say, transcription factors are required, but the pancreatic cell doesn't express those, and so therefore the hemoglobin gene is not switched on in its expression. We'd see the reverse situation in the immature red blood cell. The correct transcription factor population to express the hemoglobin gene would be present, but we'd be missing essential transcription factors that are required to activate the insulin gene, right? So our proteins that bind here at yellow and red that were expressed in the pancreatic cell are not expressed in the red blood cell, and so we don't see the insulin gene being switched on, right? So regulatory transcription factors interact with RNA polymerase and the general transcription factors at the promoter and can activate expression of specific genes in the genome aside from all the others, right, and regulate their expression. Now, one thing that it's important to note here is that we're typically not talking about one gene being regulated by one transcription factor. And if you think about it, that makes sense. It wouldn't really work to have each gene regulated by a specific transcription factor. If we've got 15,000 genes we have to regulate, we'd have to make 15,000 unique transcription factors. Pretty much our entire genome would be made up of regulatory transcription factor genes, right? That wouldn't work. So what we see instead is that it's a specific combination or set of transcription factors that have to bind to their regulatory sequences in order to activate expression, right? So, and this is referred to as a combinatorial code of transcription factors rather than unique transcription factors binding unique genes to regulate their expression. So as we saw here, this gray transcription factor, whatever it is, was actually in common, right, between the two genes that were being regulated. The insulin gene and the hemoglobin gene both had binding sites for that specific transcription factor, but for the insulin gene it was combined with sequences that bound red and yellow transcription factor, and for the hemoglobin gene it's interacting with pink and orange, right? These colors just representing different specific regulatory transcription factors that are involved in this process. So it's the specific combination of transcription factors that confers the specificity of regulation, right? And that's our combinatorial code. So this is sort of simplifying the situation. As we've seen before, we're just scratching the surface of how this regulation works. If we actually look at the real promoter region of the human insulin gene, this is much more representative of what we would see. Here's our +1 nucleotide, which we know is our first nucleotide that gets transcribed. Here's our TATA box in the promoter. We know that's at about -30-ish from our +1 nucleotide. What we see in the insulin promoter, and this is fairly typical, is a large region with many binding sites for transcription regulators, and they all pretty much have to be expressed in the correct time frame in order to activate this promoter. Now, they might not all bind at the same time, but specific sets of them would have to bind all at once in order for us to see RNA polymerase efficiently recruited and transcription of this gene taking place. Now, this highlights one feature of eukaryotic transcriptional regulation that's not typical in prokaryotes, and that's the fact that these regulatory sequences, what are often called enhancers, because again, typically we're talking about activation of gene expression rather than repression. These sequences in the gene or in the gene region that these transcription factors are binding to often are quite some distance away from the promoter. And so whereas in prokaryotes we saw that the regulatory sequences, for example the cap binding site and the repressor binding site, the operator, were right next to the promoter, and we saw direct interaction between those transcriptional regulators and the RNA polymerase complex. In eukaryotes, very frequently what we see is what's called promoter distal enhancers. So not sitting right at, promoter distal, not sitting right at the promoter and interacting with RNA polymerase, which would be bound somewhere right here, right, along with the general transcription factor complex, but some distance away. I mean, this regulatory sequence here, the VNTR, is over 300 nucleotides away from the promoter. Sometimes we can see regulatory sequences that can be thousands or tens of thousands of nucleotides away. Well, how does this transcriptional regulator that's so many hundreds or thousands of nucleotides away interact with this transcription factor, or sorry, this RNA polymerase general transcription factor complex at the promoter? Typically what we see is a sort of looping effect. So if here's our +1, what we would see, so here's our gene, what we would see is that that distal enhancer site would be brought close to the promoter. So this would be our VNTR over here that's being bound by this pure 1 transcription factor. And so it would be brought by a looping of the DNA into contact with our RNA polymerase enzyme that's bound here at the promoter. So it doesn't matter that it's hundreds or thousands of nucleotides away because it just gets looped around. The other consequence of that is that it can be actually displaced. You can experimentally take an enhancer sequence and move it further away, move it closer, flip it around so it's backwards. It doesn't really matter. The looping will bring that sequence into close approximation to the promoter site, and it doesn't really matter what orientation it's in. So promoter distal binding of transcription factors to their regulatory sequences in the gene is far more common in eukaryotes and rare and sometimes not even observed at all in prokaryotes, depending on the specific species here. Okay, so this really nicely illustrates that combinatorial code that we were talking about. And so we do see these kinds of direct interaction effects between these transcription factors and the RNA polymerase complex at the promoter. That does occur. But one thing that we also see, and that's actually more common in transcriptional regulation in eukaryotes, is that it's not a direct interaction of a regulatory protein with RNA polymerase and the general transcription factor complex. Instead, what we see is regulation of chromatin structure instead, right? Chromatin structure that in the region that the gene is located in. And so if we remember back to our discussions of what DNA really looks like in a eukaryotic cell, you'll recall that we don't see naked DNA. What we see is DNA that has been coiled around complexes of histone proteins, which are illustrated in this little cartoon diagram as the oval blobs here with the DNA looped around twice. And these form those beads on a string structures that we talked about before, the nucleosome. As we discussed before, this first level of compaction or chromatin structure then gets folded or coiled into higher levels of chromatin compaction. So first we see the coiling into the cylinder and then the folding and looping out into the higher order compaction of chromatin that we see, all the way up to that extreme level of condensation or compaction that we see during cell division. So for example, during mitosis when we look at a metaphase chromosome. So the chromatin structure can change, right, by changing the coiling and looping and compaction level of the DNA. So if we think about it, there are multiple states that chromatin can be in, and they kind of range from, a state where the chromatin is in a pretty open and uncondensed form to a form where the chromatin is more highly compacted and closed down. And so these two different versions of chromatin are referred to as euchromatin for the open chromatin state and heterochromatin for the closed or condensed chromatin state. Typically, euchromatin is associated with regions of the genome that have lots of genes in them. So these are gene-rich regions with high levels of expression of those genes. Whereas heterochromatic regions are typically gene poor, not a lot of genes present, and very little transcription going on there. Typically this is the state of chromatin that we see, for example, at centromeres and telomeres, which, as we've talked about before, are really important structural regions of the chromosome that, in the case of the telomere, confer protection from degradation. We saw that capping by the telomere protein complex. The centromere, that site where the kinetochore gets assembled during cell division, these are typically associated with heterochromatic regions of the chromosome and not many genes present, right, and a very compacted chromatin state, even when the cell is not dividing. If we have a region of the genome where we've got genes and they have to be expressed, it makes sense that the chromatin would be in a more open state where RNA polymerase could then access it, right? But what we see in eukaryotic cells is that this open chromatin to closed chromatin state of the DNA is actually dynamic, and it can change over the life of an individual cell depending on conditions that are going on, and it can also change during the development of a multicellular organism, such that different chromatin regions of the genome are being compacted or condensed down and other regions are not, right? And so this is a mechanism by which we can control access of RNA polymerase to specific genes or gene regions, and it's very commonly used as a means of regulating gene expression in eukaryotic cells. Now obviously prokaryotic cells don't have chromatin, remember? They supercoil their DNA as a mechanism for compacting it, whereas we associate our DNA with these histone proteins and other chromatin proteins to coil it and condense it so that it's regulated and organized and compacted within the nucleus. So we're going to talk about two different ways that chromatin structure can be changed and is typically changed in eukaryotic cells. And the first way involves actual modification of the DNA itself. And we referred to this way back at the beginning of the course when we talked about methyl groups as functional groups. We talked about the fact that we'd come back to this at the end of the course and see that covalent addition of methyl groups onto DNA nucleotides themselves would be associated with gene regulation. And so here we are. The other main mechanism by which chromatin structure can be modified involves modification of the histone proteins that the DNA is wrapped around. So these are fundamentally two different types of modification. In the case of DNA methylation, we're actually talking about covalently modifying the DNA itself, whereas here we're just modifying the proteins that the DNA is wrapped around. So let's talk about DNA methylation first. So one thing we need to know is that methylation is typically taking place on cytosine nucleotides within the genome. It can occur on other bases as well, but typically we're talking about cytosine methylation. And most often what happens when a gene region has undergone DNA methylation is that we see long-term or even permanent silencing of that gene. In other words, that gene is taken into a heterochromatic state, closed chromatin state, and really made unavailable for transcription because of that. Now, why would addition of a methyl group onto a cytosine base in the DNA cause the structure of chromatin to change? Well, it doesn't do it directly. So here we see sort of a diagrammatic representation of a methyl group that's been added on to a cytosine nucleotide in the DNA. And there's nothing about that methyl group that changes the interaction of the DNA with the protein. Instead, what happens is that it serves as a binding site for recruiting proteins that can come in and remodel the chromatin structure itself. So it will recruit other proteins that will then alter chromatin structure. So not a direct effect, but an indirect effect. But pretty much this is generally irreversible. Right? So once we methylate this DNA region, we don't unmethylate it. It stays methylated and it stays silenced by this permanent alteration of chromatin structure that we see going on. So when would we want to permanently silence a gene? What type of a gene or genes might we see a cell wanting to shut down permanently and never hear from again, essentially never express again? Well, there are a couple of scenarios that we could imagine. If we go back to our example of our different cell types, remember we talked about pancreatic cell versus an immature red blood cell. Is this cell ever going to make hemoglobin? No, it's never going to make hemoglobin. Is this cell ever going to be a lung cell? Is it ever going to be a neuron? Is it ever going to be a muscle cell? No. It has a specific cell fate that it has taken on, which is an endocrine and possibly exocrine, depending on what pancreatic cell we're talking about. But it has a specific function that's associated with it, which requires a specific set of genes in the genome to be expressed. All those other cell type specific genes that are not required for this pancreatic cell to function, they can be shut down, right? So one category or class of genes that we can see DNA methylation permanently silencing is cell type specific genes in other cell types other than the cells that need to express that gene, right? So muscle versus lung versus bone versus neuron, whatever. That's one class of gene that we might want to permanently silence. The other class of gene that very frequently DNA methylation is associated with permanently silencing is a class of genes that's functioning early in embryonic development. So if you think about the massive waves of cell division that have to happen during early embryonic division, it's really incredible in scope. We start with one fertilized egg, and we have to expand that one cell out into millions and millions and millions of cells that will then differentiate and take on specific cell fates. Well, there's a whole set of genes that drive those rapid rounds of cell division early in embryonic development. Once we get past that early stage of zygotic and early embryonic development, we shut those genes down, right? We shut them down because never again in the life of the organism are we going to have such rapid rounds of cell division taking place. So much mitosis going on as we see early in embryonic development. And in fact, reactivation of those genes, so if methylation goes awry and isn't maintained in those genes, is a genetic change that has been associated with some types of cancer. So we can see reactivation of those zygotic genes and rapid cell division being initiated as a result of that. But typically, those classes of genes would be permanently silenced following that specific stage of development. All right, so that's DNA methylation, permanent silencing. What's the other form of modification involved with our histone modification? This is a more dynamic form of regulation. This is reversible. So we have enzymes that work in both directions here and confer this short-term dynamic, very responsive to changing conditions form of regulation. And what we're talking about here, again, is modification of the histone proteins that DNA is wrapped around. So if you remember earlier in the semester, we looked at this more structurally accurate representation of a nucleosome. Here's our DNA wrapped around twice around the exterior, and here's our complex of histone proteins in the middle. And remember that I pointed out to you these little tails that project out. So these are N-terminal domains that sort of protrude out from the nucleosome complex, right? And these are the parts of the histone proteins that get modified by covalent modification. So we see specific types of functional groups being added on and then removed from these little N-terminal tails, right? And so what types of modifications do we see? Well, we can see phosphorylation, which we talked about before in terms of enzyme function and enzyme regulation. We talked about reversible phosphorylation as a mechanism to do that. Same thing here, nothing new. We can also see methylation coming into play, right? So addition of methyl groups onto these N-terminal domains of the histone proteins. What we're going to talk about is actually the most common modification that happens, which is acetylation, addition and removal of acetyl groups onto these little N-terminal tails that project out. So typically, when we have enzymatic modification of histone proteins such that an acetyl group is added, so that would be called acetylation, that is associated with less condensed chromatin. And as we've talked about before, less condensed chromatin, a more open chromatin state, is going to be more accessible to the transcription machinery, and we're going to see transcription activation as a result of that enzymatic modification. So adding an acetyl group on is going to tend to result in activation of transcription of that gene in the nearby region. So the proteins that carry out that acetylation reaction are called histone acetyltransferases, or HATs. So HATs modify chromatin such that chromatin structure becomes more open. The transcriptional machinery, RNA polymerase, general transcription factors have more access, can bind the promoter more easily, and therefore transcribe the gene. On the other hand, histone deacetylases, or HDACs, catalyze the reverse reaction, removing this acetyl group from the histone tail. And as addition of the acetyl group was associated with a less condensed chromatin state, removing it is going to be associated with the opposite, right? A more highly compacted or condensed chromatin state, which is going to lead to transcription repression, right? And so we go back and forth. We have these enzymes sort of competing with one another, right? HATs adding the acetyl groups on, HDACs removing them, obviously in a regulated fashion, and in so doing changing the structure of chromatin in a gene region, which is going to change the activation state of that gene and whether it's being transcribed or not. Now, what would cause a HAT or an HDAC to interact with this specific gene region as compared to some other? Well, very often these regulatory transcription factors that we talked about before, binding to enhancers, these could actually be recruiting these HATs and HDACs to this gene region. So some of these transcription factors could actually not be directly interacting with RNA polymerase, but actually binding to a HAT enzyme and recruiting it to a promoter region to change chromatin structure. And so these are chromatin modifying enzymes that are modifying chromatin by covalently modifying the histone proteins that DNA is wrapped around, right? Now again, what specifically does acetylation or phosphorylation or methylation of these N-terminal tails do to alter the structure of chromatin? Well, we think nothing directly. Once again, what we see happening is that when we see these modifications being added, we recruit these chromatin remodeling proteins to come and sit down and cause the chromatin structure to be organized in a different way, to go from a more highly compacted state to a less highly compacted state, changing the spacing between these nucleosomes, changing the level of coiling and condensation, etc. And so this is very much an indirect effect, not necessarily a direct interaction of proteins with RNA polymerase to influence the initiation of transcription, but to more globally change the structure of DNA such that it either favors the ability of RNA polymerase to come and bind and express that gene or not. Very different from what we saw in prokaryotic cells.